Welcome to session 3 of the Fenner Conference on the Environment. This session is a panel session and we will begin with Dr. Karen Sommerville, recapping this morning's first 2 sessions and sharing the results of our poll. Thanks very much, Karen.
Thanks, Amelia. Okay, so we've heard today that plant species can fall into the category of exceptional for a number of reasons. They might have seeds that aren't suitable for seed banking at all, or that are relatively short-lived in storage. They might have seeds that are very difficult to germinate, no longer produce seeds in the wild, or they might produce some other type of reproductive structure instead of seeds. All of these plant species need alternative measures for ex situ conservation. Earlier in the conference, we had some really nice detailed presentations on how to identify different kinds of exceptional species and a range of techniques that might be needed to preserve them. Some of those talks were quite technical, so if you haven't delved into these sort of topics before, then you might like to refer to chapters 6, 9 and 10, especially of the revised germplasm guidelines for an overview. For now, I'm just going to summarise the main points from each presentation as a starting point for the panel discussion. So in the opening presentation, Professor Hugh Pritchard from the Royal Botanic Gardens, Kew, highlighted the need for investment in alternative preservation technologies, and particularly cryobiotechnology. These technologies might also be needed for species with orthodox seeds, as many are proving to be shorter lived in conventional storage than expected. The development of cryobiotechnology is particularly important for preservation of species with recalcitrant seeds. That's seeds that don't survive drying. So, for example, of the 57,302 tree species around the globe, it's been estimated that around 40% have recalcitrant seeds, and almost 40% of species listed as critically endangered under IUCN Red List criteria also have recalcitrant seeds. So if we want to meet conservation targets under the Global Strategy for Plant Conservation, investment in alternative conservation strategies is essential. The next presentation was from me, and I had a reviewed how we go about identifying whether a seed is orthodox or non-orthodox. Non-orthodox seeds are those that don't tolerate the conditions normally used for seed banking. So that includes recalcitrant seeds, which don't tolerate drying at all, and intermediate seeds, which either can't be dried to the same extent as orthodox seeds or can be dried but don't tolerate storage at sub-zero temperatures. Non-orthodox seeds can occur in a range of environments, but are most common in tropical and subtropical rainforests. And the most reliable way to identify those seeds is with a series of tests comparing germination of fresh seed to germination after drying and cold storage. But there's now a key that can help to identify desiccation tolerant or sensitive seeds without that germination testing. So that presentation was followed by a video that took us through the use of the key and demonstrated how various seed characteristics can be used to identify desiccation tolerant and sensitive seeds. The next presentation was from Dr. Dave Merritt from Kings Park Science, and Dave spoke about seeds that are orthodox but short-lived regardless of the storage conditions. These seeds can be identified by running comparative longevity experiments in which the seeds are rehydrated, incubated at 60 degrees for set periods of time, and then germinated to determine the number of days needed to reduce germination by 50%. And enough data is accumulated from these sorts of experiments now to see that seeds from cool, wet environments tend to be shorter lived than seeds from warm and dry environments, and also that alpine seeds are shorter lived than seeds from the same family in lowland habitats. These experiments are really useful for giving an indication of species that are likely to need more intensive management or might need alternative storage conditions. But Dave pointed out that the P50 value from an ageing experiment doesn't always reflect the amount of time a species will remain viable in storage under dry and cool conditions. So for species that appear short-lived, it's a good idea to investigate their response to storage at a range of different moisture contents and storage temperatures. I'm just going a little out of order now, but I wanted to mention Cathy Offord's talk first on living collections as a lead into the talks on tissue culture and cryopreservation. And Kath mentioned that a living collection can take a few different forms, from potted collections like those held for the Willamite pine, to collections grown in ground in botanic gardens and arboreta, and collections grown in field gene banks or seed production areas. As an option for conserving exceptional species, living collections can provide many benefits, especially if they're genetically diverse. And those benefits include providing a source of material for translocation, for horticultural research that can inform in situ management, and to provide material for tissue culture and cryopreservation. Living collections can occupy a considerable amount of space and can require quite intensive management to keep pests and diseases under control, but spreading the collections among several facilities can help to minimise the space and handling required in one location. Where space and funding are available, living collections are a good option for initial preservation of exceptional species and can provide material to support other conservation methods down the track. And then we heard from Eric Bunn from Kings Park Science, who spoke about the use of tissue culture as another method for conserving exceptional species. Tissue culture is a technique for growing species in a sterile environment on a medium like agar that contains the nutrients and hormones needed for plant growth. Cultured plantlets can outgrow their container within 1 to 2 months, so they need to be recut and transferred to fresh media regularly. And this can make the technique quite labour intensive when multiple individuals of multiple species are cultured, though the amount of labour involved can be reduced if the growth rate is slowed by reducing nutrients and light levels and temperature. Tissue culture can be used as a conservation technique by itself to produce multiple plants for research or translocation, or as a stepping stone to cryopreservation. But cryopreservation is what's needed for long-term conservation. Eric then described some of the many complexities involved in preparing tissue cultured material for cryopreservation and the difficulties involved in recovering the material after freezing. And he then mentioned some alternative culture technologies that might help to improve the response to freezing, such as autotrophic culture. In our second session, we heard from Valerie Pence from Cincinnati Zoo and Botanical Garden. And Valerie started by summarising the different types of material that can be cryopreserved and the relative effort involved. From orthodox but short-lived seeds, which can be preserved pretty simply by drying and packaging the seeds and then placing them directly into liquid nitrogen, to tissue cultured material, which requires intensive work to develop culture protocols, prepare the material for cryopreservation, and then to recover the material after freezing. Once material is cryopreserved, though, it requires little further input of time or resources to maintain. At Cincinnati Zoo and Botanic Garden, cryopreservation has been used successfully to preserve short-lived seeds, fern spores, fern gametophytes, pollen grains, embryos, and shoot tips. And Valerie noted that the development of protocols for cryopreservation is the most time-consuming part of preserving species this way, and that innovations to make the process more streamlined and more broadly applicable were needed. She also noted that the importance of collaboration among multiple institutions in this process and described the Exceptional Plant Conservation Network website, which has information now on over 775 exceptional species, and where it's possible to add information on new species, as well as contact details for people working in the field to make it easier for people working on similar taxa to connect and collaborate. Bryn Funnicotter from Curtin University in Kings Park Science then provided an overview of cryopreservation in Australia, noting that the first trials of cryopreservation in Australia began in 1992 with the threatened Grevillea scapigera. And since that time, 145 plant species have been successfully preserved using seed axes, spores, protocorms, callus tissue, and shoot tips. Bryn noted that tissue-cultured shoot tips are a good source of material for cryopreservation because their small size allows good penetration of cryoprotectants and they produce true-to-type plantlets. He outlined a few different methods for preserving these and then gave an outline of all the steps involved in a method known as droplet vitrification. Innovations that have improved cryopreservation success for this procedure include the use of a vacuum to improve the infiltration of cryoprotectants and the use of small pieces of metal mesh to aid handling of the shoot tips. Some species, though, have proven really difficult to preserve this way and show slow or abnormal growth or oxidative damage following cryopreservation. Jayanthi Natarajan from the New Zealand Institute for Plant and Food Research took us through the procedure for extracting and cryopreserving embryonic axes. And this involves careful extraction of the root and shoot axis from the whole seed when larger seeds are used. Embryos collected at full maturity are more amenable to desiccation, but oxidation can occur rapidly in some species when the axis is extracted. Vacuum infiltration can greatly improve the uptake of cryoprotectants, as it did for shoot tips, And then Jayanthi gave us an example of Syzygian mare, which was an example of one species that's proving very difficult to cryopreserve because of a range of issues, including a narrow window for collection of fresh material, large and variable embryo sizes, and differences among populations in seed maturity. And then Jayanthi showed us a great video showing the steps involved in excising embryonic axis from this species and ways to avoid oxidative stress. And finally, we heard from Daniel Ballesteros from the Royal Botanic Gardens Kew, who focused on the cryopreservation of fern spores. And he noted that spore banks were the preferred method for ex situ conservation of ferns because they allow high levels of genetic diversity to be preserved in a small space with minimum technical requirements. Oops, forgot to change slide, sorry about that. And there are two main types of fern spores. There are green or chlorophyllous spores, which have a short lifespan due to the chlorophyll content, and non-green or non-chlorophyllous spores, which are longer lived. Danny mentioned that the spores are tolerant of desiccation, a bit like orthodox seeds, but they dry very quickly and they don't like to be over-dried. So drying for just 1 to 3 days is enough for green spores and 5 to 7 days for non-green spores. The dried spores can be stored for 1 to 10 years in a fridge or freezer, but will have a longer life if stored in liquid nitrogen. And Danny then went on to give examples about how fern spores could also be used in fundamental biological research to better understand how seeds respond to storage. And that gives us our starting point for this afternoon's panel session. And what we'd like to do now is talk about how we can use all these different tools to conserve our national plant treasures. But before we start that, I'd just like to go over our poll results for the day. Okay, so our first poll question was, what is your motivation for attending the Fenner Conference Day? And we had 89 people responding. 25 are actually working on exceptional species already. 3 intend to work on exceptional species in the next 5 to 10 years. 17 were interested in different methods of storing germplasm, but most people were just interested in plant conservation in general. Can we go to the next slide? Okay, so the second question was, what are the challenges in conserving exceptional species for you? And we had, and again, 89 people responding, and identifying which species are exceptional was the biggest problem. Access to resources was another problem. And issues with germination and propagation were a problem. I can sympathise with that. And then difficulty finding or accessing relevant information was an issue for 21 people, and lack of capacity to address exceptional species was an issue for 13. Our next question was, where do you find information on exceptional species? And we had 57 people responding to this one. Most people find it in peer-reviewed journal papers, 35 people from formal guidelines like the Millennium Seed Bank Conservation Standards or the Plant Germplasm Conservation in Australia guidelines, 32 people from databases like Kew's Seed Information Database, 33 from local or regional contacts, and 31 from personal contacts. And the last question was, what would you like to achieve in the next 5 to 10 years? And 57 people again replied, and to this we had 27 people wanting to start to bank and conserve exceptional species, which is great. 43 people want to get more species into ex situ storage, which is also wonderful. And we had 30 people wanting to examine existing data to understand which species need alternative methods, 25 wanting to join a regional network for better support and coordination, and 27 looking for resources and funding to support conservation of exceptional species. All right, thank you very much for that. So that leads us to our panel session, and our facilitator for this afternoon's session is Dr. Cathy Offord. Cathy is the Manager of Germplasm Conservation at the Australian Plant Bank and has had a very long-term interest in ex situ conservation of rare and threatened species of all shapes and sizes, using seed banking, tissue culture, cryostorage, and living collections. So she's a good person to be guiding the discussions this afternoon. Thank you, Cathy.
Thanks, Karen. And that was a great summary. And good afternoon to all the participants. There's 132 of you online at the moment, which is fantastic. Look, this afternoon, in the previous 2 sessions of this webinar, we've had a really good introduction to the techniques that we can use to identify which species are exceptional and the techniques that we can use to conserve them. So now we're going to put all that together in this panel discussion. We've brought together a panel of experts to discuss a number of questions that often arise when we're working with exceptional species. And these sorts of questions will include why do we need to think beyond the seed bank for ex situ plant conservation? What are the challenges for conserving exceptional species? What have we already achieved? And there is quite a lot that's been achieved that we saw this from the presentations. What's working well, what isn't? How, and this is a really important one, I think. How do we collaborate, and how are we going to form really workable partnerships that's within our own organisations and across organisations? How relevant is the conservation of exceptional species to different types of conservation, such as ecological restoration, and then where to for the science of exceptional species? So as Amelia said before, if you've got questions, can you put them in the Q&A? And we will try and get to them during this session. If not, we'll address them later after the panel session. So our panel members today have a wealth of experience with ex situ conservation in the region. And I'm going to get each of them to introduce themselves and their interests and their experience with exceptional species. So if I could get all the panel members to turn their cameras on. Thank you. And I'm going to go to you first, Linda Broadhurst.
Thanks, Cathy. This is my first time at the meeting today, so I just want to acknowledge the traditional owners of the Ngunnawal and Ngambri land that I'm sitting on today. So I work at CSIRO. I'm currently a senior scientist there. I was until recently the director of the Centre for Australian National Biodiversity Research, but I've recently stepped back into science. And I've got about 20 years experience applying molecular and ecological tools to enhancing native vegetation. And with that comes the sourcing of high genetic quality seed to ensure that our restoration is underpinned by this very broad evolutionary base that's going to be able to cope with change as we go in a warmer, drier climate. Thanks.
Fantastic. Thanks, Linda. I'll go to you, Karin van der Walt in New Zealand.
Thank you very much. I'm Karin van der Walt. I am from Ōtari Native Botanic Garden in Wellington, New Zealand, and my role is conservation and science advisor. So at Ōtari, we started a small bank, a small plant bank, sort of a 1% of the Australian version. And there we've got facilities for conventional storage and cryopreservation. So that is our big focus. We do a little bit of tissue culture, but as you've heard, it is quite challenging to incorporate tissue culture. And then, of course, using the living collections at Ōtari to contribute to the conservation.
Great. Thanks, Karin. Lydia Guja in Canberra.
Hi, everybody. Thanks for the opportunity to be part of the panel today. So my name's Lydia and I'm from the Centre for Australian National Biodiversity Research, which is a joint venture between CSIRO and the Australian National Botanic Gardens. So I sort of wear two hats. One is as a seed biologist, where I look at seed banking and seed germination in a science part of my role, and the other half is managing the National Seed Bank. And in the National Seed Bank, we do seed banking, but also organisationally, we're part of Parks Australia, which brings with it a heap of exceptional species. We've got a lot of threatened ferns and subtropical and tropical species that we're interested in banking. So a real diverse range of interests for our group.
Great, thanks, Lydia. Nathan Emery in New South Wales.
Hi everyone, my name's Nathan. I'm a restoration biology officer at the Australian Plant Bank, part of the Australian Institute of Botanical Science. My research is a combination of plant ecology, seed biology and restoration. I've worked with worked on a few difficult to propagate species such as Actinotus and a number of threatened Petunia species. And I've also worked on developing several plant translocation programs for some of these threatened species, mainly Petunia, but also a couple of other threatened species under the Saving Our Species program. And I currently also work on the seed biology of several threatened ecological communities in New South Wales.
Great, thanks, Nathan. Let me see, Damian Wrigley, back to Canberra.
Thanks, Cathy. I'm Damian Wrigley. I'm National Coordinator for the Australian Seed Bank Partnership, but I'm also the National Focal Point for the Global Strategy for Plant Conservation for Australia and the Oceania Representative to the Plants Committee for CITES. So I guess I'm very much a plant science generalist. I have been working in biodiversity conservation in various levels of government for more than 10 years. And in this role, looking after the Seed Bank Partnership for over 4 years now. So I'm constantly learning every day. I always have that imposter syndrome when someone calls me an expert, but I work in partnerships and scientific networks for quite a while. So I guess that's where my expertise is.
Thanks, Damian. Gibson Sosanika in Papua New Guinea.
Hi, everyone. Thank you. I am a lecturer at the Papua New Guinea University of Technology at the Department of Forestry. I've been working with villages, the landowners, for 10 years. We've been conserving 50 hectares of dynamic plots. We conserve about 500 species, more than 500 species, and tagged about 350,000 stamps under the CTFS protocol for plant database management. So by just protecting the forest, it has given us some opportunity to work with the Kew Gardens in part of Australia Crop Trust, so that we look at the seeds of New Guinea, because it's one of the first of its kind in Papua New Guinea will be established. And I'm working on that as part of the university as well.
Excellent, thanks, Gibson. And last but not least, Karen Sommerville in New South Wales.
Hey everyone, I think you've heard enough from me today. I lead the Rainforest Seed Conservation Project at the Australian Plant Bank. So I've been working in different aspects of plant conservation since about 2003, going from salt marsh to terrestrial orchids and then to rainforest. So I've had experience in a range of different aspects of ex situ conservation, from tissue culture to seed storage and seed research, and even mycorrhizal fungi. So it's quite a broad range, makes me a little bit of a jack of all trades, but it gives you some interesting insights into different aspects.
Thanks, Karen. Great. Thanks for introducing yourself. And there is, there's a vast range of experience in this panel. So yeah, let's tap into that. So the first question, it's to all of the panelists. So why do you, or your organisation, why do we need to think beyond the seed banks?
We just have so many species that are showing up to fall into that exceptional category now, much more than I think than when we started out, even in Australia, even though we have such dry habitats generally in the country, we still have a great many species that fall in the exceptional category, even those from dry habitats, which are proving to be shorter lived. So yeah, I think it's essential that we start working harder in this area.
Yeah. Anybody else want to come in on that one?
I think for us here in New Zealand, another problem with seed banking is you need some way to quantify your viability before being banked and into banking. And very few of our native species, we actually even know how to grow them. And when we start off trying to grow them, often we come across some dormancy issues or just challenges in that way. and figuring that out can take quite a long time as well. So sometimes it's reverting back to living collections and actually just propagation and then look at seed storage.
Yeah. Any other perspectives?
So
Linda?
Cathy, yeah, I think that both Karens have made really good points. And I think, I'm stepping back, I'm stepping out of a seed bank almost, because I don't actually work in a seed bank. But for me, this is just the first step, is the banking. It's the getting back to the environment that's actually the critical success of a seed bank. And there are all these issues and all these species that we're really challenged to do that with.
Yep. Thank you. Well, look, I might move on to the next one. This one's for, I might give this one to Lydia, actually. How do you decide on the priorities for conservation of exceptional species when I think everybody to some extent has this issue with where the resources are finite. So, how do you prioritise?
It's a really good question, Cathy, and I feel like it's a hard one. I'm sure others might have things to add, but I guess just starting with the species that are listed, they're on that list for a reason. They've been assessed, we know they're at risk, but you're right, resources are often finite and we can't necessarily bring all of them into ex situ conservation. But I think that's where we can take almost a bit of a triage approach and look at the risks in the environment, and then also in ex situ conservation, the technologies or the knowledge that's available to us at the time. So some species we might kind of know a bit more easily stored. Others we might know that we're up against a research and development challenge where we need to work out how to get that species secure. So I guess there's a lot of criteria that we can use to prioritise, and I think prioritisation itself is important to do, but maybe sometimes we get a little bit hung up on looking at different prioritisation lists and maybe concerned if they don't align. But if we're using different criteria, we're going to get different answers. As long as species have come up for some reason and there's a need to work on them, then I think it's a good reason to continue.
Excellent, thank you. All right, so Nathan, so how can a variety of ex situ methods support in situ actions like translocations? So, can you give us your experience in taking out of the seed bank or the those tricky species that you've had to deal with over the years?
Yeah, sure. It's certainly a complex question, and it's got a complex response. I guess right off the bat, it's important to allow at the start of such programs to investigate the ex situ strategy and consider what works and what might not work, because I think that will determine what path you're, when you're thinking about translocation, how you'll do your planting design and what form your collection strategy will take. So ideally, using seeds is the most cost-effective method to grow plants, to produce plants for translocation. But in the cases that I've worked on with Basunia, it's taken many, many years to even be able to collect enough seeds, understand how best to germinate them and grow them for returning them back into the wild. So, we've looked into other ex situ methods which have been critical to achieving our targets that we and our stakeholders agree to. So we've looked at vegetative cuttings, propagation, tissue culture to produce seeds, plants for seed orcharding in the nursery and for translocation. And it's had several benefits to doing this multi-strategy approach. So we're able to produce plants to grow on in the nursery, to look at things like the reproductive system, to understand if this is going to limit our ability to collect seeds from these species in the wild. We can also collect seed to study more about the seed biology and to germinate or how to germinate these tricky species like Basunia. And it's also given us additional plants which we can then put back into the wild and study how plants from different or grown from different propagation methods survive under a translocated setting. So, you know, it's helped us solve some of these missing puzzle pieces for Petunia in particular, not all the pieces. We've still got quite a few more to figure out and wrap our heads around. But, you know, because of being able to use this multi-strategy approach, we're able to know more about the reproductive biology. We know how to propagate plants, and we know that the plants we are able to grow from seed are actually more robust in our translocation than, say, cuttings propagated plants.
Great, thanks, Nathan. I'm just wondering, just following on from that, like how can we better integrate ex situ and in situ conservation? Is there a, I mean, anybody can chime in on this one, but, you know, because they're not, you know, they're tied together inextricably, but we sort of tend to separate them out into two areas. But how can we do this better to get better outcomes in the wild?
Yeah, I guess I can start on that.
Only Nathan.
Yeah, I think it's really important to be able to integrate the two of these together. You know, for example, if we can understand more about the germination the requirements that a species need in order to germinate, you know, the temperature, the moisture, the fungal associations for some species, then we can use this knowledge to better predict things like recruitment events and things in the wild, and be able to better manage those populations in situ and able to, I guess, prioritize those populations which may be at risk of say, recruitment failure, for example. So yeah, I think there's some great ways that the two can work together as one. Okay,
thanks. I'm just wondering there's differences in scale, you know, for sampling, you know, for different purposes. So, you know, for research or for translocation or for, you know, broad scale restoration. How do we better integrate? How do we do this better and integrate using the vastly improved genetic techniques? You know, they've come ahead in leaps and bounds just in the last, you know, 1 to 2 years. How can we use those techniques to better integrate and get the population, capture more diversity in our collected populations? I might give this one to you, Linda.
Thanks, Cathy. Look, I know it seems like there are vast differences between, you know, what you're collecting for a conservation seed bank and what you're collecting for ecological restoration, but there are a huge amount of synergies. You know, you're both working with a fundamental unit of seed, you know, and you might have a rare species, so you're going to collect as much as possible. Well, for ecological restoration, that's often also the case. Both come up with the same problems. You know, you go out, there's no seed, you're too early, you go out, you're too late. You go out that season, it's been burnt by a fire. These are, you know, common problems between the two areas. So I think it's really important not to think of them as vastly different. I think there's lots of synergies, and I actually think that what conservation seed banks do could be really informative into the ecological restoration world in terms of germination, propagation, those sorts of things. In terms of genetic diversity, you know, when you're with a rare species, you're pretty much collecting everything because that's what you're working with, and you want to capture all that. For ecological restoration, as good as that might be, you just simply can't afford to collect everything. You don't have the time, you don't have the resources. So you do have to kind of triage what you're collecting a bit. And so we often go collect from as many populations as you can, and from, you know, somewhere between 10 to 30 mother plants, so that you are really broadly capturing whatever is available at the time you're in the field. And sometimes we'll say, go back to the field a second time, which I think conservation seed banks do as well. You know, you may visit a site more than once because you are picking up different bits of diversity every time you go.
Yep. Thanks, Linda. Look, I'm just going to pick up on a couple of questions that came up this morning, and just follow those down. Just in terms of how we collect and what's actually sort of missing from our collections at the moment. So do we have a sense of what's happening with the conservation of fern spores in Australia? Is that, Lydia, is that something you can?
Yeah, I think it's looking like a really critical and emerging area. I guess I can sort of talk for Tom North, who's my colleague and the author of Chapter 13 in the Germplasm Guidelines. He and Danny and others looked at fern spore conservation, and they've got a really nice summary there. But I guess for us and the AMBG, our interest came from our work with Parks Australia. So Norfolk Island is one of those places, and a really good example where there's a huge number of endangered species. It's something like 45 listed species, and I think approximately 15 are ferns. And so there's a need to conserve those. They're very range restricted, but they're also ecologically quite important in that landscape. And so that's something that got us interested in this and started us off on this journey of trying to store and understand spores. And there is a lot we can do, and I think we've got a really good sort of infrastructure already across the country with the different seed banks and storage facilities that we have. And so it's really something that we should be able to do together and start to focus on those species. And perhaps some of it is going to require a better understanding of that plant group to begin with. I think we've had a lot of good, we have a good understanding of our flowering plants, but maybe we don't have as good an understanding of all of the species of ferns or where they occur, and whether there might be more that might need to be listed.
Okay, just from the chat, I've got a comment here that RBG Victoria is developing a fern spore bank, and they've got an aim to store 35 Victorian species by next July. So that's great to hear.
That's awesome.
So obviously something that people are putting their minds to, as we're all starting to grapple with exceptional species, so that's really great to hear. So the other thing that came up this morning, also from RBG Victoria, was Tom May was asking that given that most plants have some kind of beneficial symbiotic relationship, like mycorrhizal association, mycorrhizal fungi, can we see ourselves also trying to preserve those and understand those relationships and add those into our biorepositories? And we're already doing this for orchids to some extent, but could we be looking at that more generally for our species?
I think it's a good idea to start looking at it, but it's quite complex. Finding the right symbiont in the first place and then working out the best way to store it. I think it's definitely worthwhile for the species that are absolutely dependent on those. But we might need to get a whole bunch of students on board to look at the ones that aren't, that are beneficial but not essential.
Yeah. So there is an emerging research theme that we saw in during the speaking program Monday and Tuesday, with trying to understand the seed microbiome and what the function of the microorganisms that we're finding. We used to think of them as contaminants and throw them out, but now they actually could be useful. So I think that is an interesting area of research, but with an aim to conserving and using those symbionts. Well, I might just ask Gibson, in Papua New Guinea, what's the situation with seed banks or germplasm repositories? What's happening there with exceptional species?
For Papua New Guinea, the seed bank concept is sort of new idea. Mostly it is deal with the commercial plants, and there is no protocol of doing that for seed exchange programs or seed sharing data on that. I just started, and I collected about 100 species, and in total botanical survey that people have done it, the record shows that there are about more than 5,000 species expected to be discovered in PNG, but it's a lot of problem with doing research and all that. So it's kind of starting, but with the CTFS protocol that we set up, the CTFS 50-hectare forest plot, we documented about 500 species. And on behalf of my work, I've seen that those species needs to have some seed collection on them. And with this conference and this medium, it's already a good connection of sharing of ideas on how the species have been seen between the orthodox and Tasmanian seed, and more study will give more research and more work to be done.
Great. Well, yeah, I'm hoping that we all can reach out to each other, after this conference, and share our knowledge and share our techniques. I mean, we'll have to do that virtually for a while, but hopefully we can have some more sort of face-to-face contact and share our knowledge and techniques.
Yes. There was a training done with Damian and the team in Australia, and I've gained some experience on that. The problem is with the plans to continue collecting the seeds and in them in Tasmania.
Yeah, Damian, do you want to sort of take us through what's happening in the region with training and outreach?
Can you come again on that question?
Oh, sorry, I was just going to ask Damian if he could tell us what's happening in the region with
yes.
Yeah, with outreach.
Thank you.
Thanks, Kath. So I guess some of the training that has been rolled out in the region has really come from Millennium Seed Bank Partnership from Kew Gardens. They've done a lot of training with their blue drum kits for the field and gone out to different countries, including New Zealand for myrtle rust, and some of our partners went over and helped with that a couple of years ago. I think discussions like last night's one are going to be really important for building that kind of training and facilitation across the region. There's also crop wild relatives work that the Crop Trust has been doing, and so that's the project we collaborated with Gibson on, bringing out a couple of people from so Gibson, another member from Indonesia, to do some training at Kakadu, and also go down to Horsham with Sal Norton and do some training in the lab. So it really comes down to the projects that have some funding available for it. So it's taking advantage of what we can through these networks and sharing information and opportunities, I think.
Right. I have a question here from Diane from Indonesia, and she's asking what our challenges and what are the benefits from integrating the seed banks the way that we have in Australia under the the partnership, because they don't have that in Indonesia at the moment.
Yeah, thanks, Kath. Thanks, Dan. I mean, the challenges we have is resourcing. So even though a lot of our major conservation seed banks are run by either local, state or federal government, they still struggle for resources like any other sector, whether that's funding or people. So I think some of those limitations can certainly be an issue. Also, some of the limitations of what you can actually do with your facilities, whether or not you're allowed at your level to reach out and join in some of those discussions like we had in the seed network discussion last night. I think also for us, it'd be ideal if we could reach out into those broader networks of conservation seed banks across Australia. But again, for us, it comes down to resourcing and being able to coordinate that. And I think projects like Project Phoenix, that's been trying to bring people together and improve conversations and networks across Australia's seed sector are really important. There's still obviously a long way to go, but I think things like that and healthy seeds that AMPC has been working on, these are really important conversations for us as a country to be able to bring together everybody that's trying to work in this sector.
Great, thanks, Damian. So I might go back to Karin. We're talking about regions and networks, but sort of it's also good to understand what our species are doing, because species don't stop at the borders. and we share a lot of our flora, you know, across the region. And Karen, I'm just wondering if you could sort of summarise the work that was done a few years ago in understanding the seed storage behaviour of the South Pacific region, and particularly what we know, but what don't we know?
Yeah, so we collated all the species from a number of different countries around the Pacific, and we found that we got up to about 1,503 genera across the Pacific, many, many more species, of course, and about half of those had never been studied as far as seed storage behaviour goes. So there's a lot of scope there for research and for better understanding the flora and whether or not we can seed bank them. And the good thing about what we found was that a lot of those genera are shared across at least one other country. So there's a lot of potential for collaboration between countries, or for us, if we have a genus that appears in some other country, to share the information that we found so that we can give a start to somebody who's just starting to work out in that area. So yeah, there's lots that we don't know, lots of potential for research and lots of potential for collaboration.
Yeah, okay, great, thank you. All right, I might go over to New Zealand, to Karin, and just ask you, you know, we have a bit of a focus on rainforest species in New South Wales because it's a very diminishing flora, and we have a problem with myrtle rust, which is wiping out our species, and I know it's now in New Zealand. Just wondering if you can sort of take us through what's happening there with those sorts of species, maybe those that are affected by myrtle rust or potentially affected by myrtle rust, and species that are important to First Nations people.
Sure. Yeah, thanks, Kath. Yeah, as you said, we've had myrtle rust now since 2017, so really starting to see the impact on a species level now. So in New Zealand, we've got about 32 native Myrtaceae species, which I know is not a lot compared to Australia. But the main thing is we don't know a lot about those species, as I said earlier. One of our problem children has come up in a couple of discussions already, and that's Zyzigium myri or swamp myri, the same as lily pilly. So it's the challenges has been well discussed by Jahanti. So we've been working on that. And the race is on now because we've seen the impact from myrtle rust on Zyzigium species now. So we can see that it's destroying the fruit and the flowers. But then another example we've got, which is actually quite a nice one because it summarizes some of the discussions we've had already, is a tree called Bartlett's rata. So it's a Metrosideros, Metrosideros bartlettii, and it occurs right in the northern tip of New Zealand. And there's only 14 of them left. So they are obviously Myrtaceae species. Their seeds will be orthodox if we look at the genus, but the problem is they no longer reproduce in the wild. They are represented in cultivation throughout New Zealand, but the genetics lack. So most of them are from clones. I think it was 98% of the trees in cultivation are actually a clone. So what we've done at Ōtari is we use our ex situ collection. So that's integrating the in situ and ex situ collection. We use those trees. So the trees in cultivation have been sequenced. So we do know which ones aren't related to each other. And we went on to test the pollination strategy for this tree. So having a plant or a tree close by. So I just mentioned Bartlett's rata grows in Te Paki. That's a very long way from us. But at Ōtari, we can go out every day and look how the flowers develop. When is the stigma ready? When is the pollen ready? And we could test the pollination strategy for this tree. And through this, we confirmed that Bartlett's rata is self-incompatible, which actually makes the problem even worse because now we need those 14 trees unrelated to each other to produce viable seeds. And then what we also did from there is we banking the pollen because the trees in cultivation are not flowering at the same time. They're slightly out of sync, not hugely. But what we're doing is we're collecting the pollen and then we store the pollen so we can bridge not only the short-term gap, the days gap that we needed, but also in the longer term. So when you've got an individual flowering, that you can actually get the pollen from the unrelated individual. And now that we understand those techniques, we're actually going up to Tapaki, and we're going to work with the local landowners and the local iwi in that area, and we're collaboratively working by understanding the pollination. We can now apply it to those trees in the wild and solve, hopefully, the reproduction failure that the tree is seeding, and then collect the seed and store it, usually orthodox ways.
Thanks, Karin. I've got a question here from Peter Cuneo. It's also about myrtle rust, and it's what do you think the long-term options for establishing living ex situ collections are in unaffected regions, or regions where myrtle rust is unlikely to occur? Is that one way that we could conserve those species and keep them as wild species?
It's certainly something that's being talked about in New Zealand, especially, and maybe it will be applicable to Australia. So the climate modelling suggests that some parts of the South Island will be unlikely to get myrtle rust. And then they got myrtle rust. So the models were proven a little bit wrong that way. So look, there is a risk to it. And as we've said, there's certainly a place, as you mentioned in your talk earlier today very well, there is certainly a place to have these large collections, but keeping in mind the genetic diversity needed to do, to have the genetic diversity represented in 50 or 500 trees or whatever that boils down to, and just the logistics of it. But I think as a complementary method to maybe alternative as well, seed banking, if that's an option, yeah, it just increases your chances of success, doesn't it? So where it's feasible, definitely you can try it.
So you're suggesting for these species that are going, you know, some of these are literally going extinct in front of our eyes, that we should be trying everything that we can do within our resources?
Yeah, I think it did definitely not sit back and say, oh, well, that seems just too hard. I think just trying whatever you can. I think you've got some of the species that you're collecting plant material now from the gardens to do in tissue culture. So if that wasn't there, the tree would go extinct, the species would go extinct. So yeah, we know what needs to be done, but also try everything, especially if we're faced with something like myrtle rust that's progressing so quickly and we're seeing the species go extinct so rapidly. You've got nothing to lose if you can do that. So
okay, so I might actually sort of move into like the role of cryo storage and its potential. So we've heard a lot about cryo storage today, but does anybody want to comment on what needs to be done to understand? We understand some species very well, particularly agriculture, species of importance to agriculture, and that's sort of the basis of our technology. But how much do we have to do to make this a routine conservation technique as seed banking, or orthodox type seed banking is for dryland species? I'm looking at you, Karen Sommerville.
I was hoping to get away from that. I think we need to follow the suggestions of Valerie Pence and Hugh Pritchard and look into the fundamental biological aspects that affect cryopreservation, and get a better handle on those so that we can, streamline the processes at the moment. Because at the moment, it takes a lot of effort to, first of all, to get something into tissue culture and get it growing well there and develop the protocols for that. And then all these different steps in the cryopreservation process need to be optimised in order to get the material coming out well at the other end. And particularly for rainforest species, it's proving really, really challenging to get anything into cryo and come out alive. So we really need to invest a lot more research into the fundamental biology of the species so that we can get some better results out of that.
Okay, thanks, Karen. Does anybody else want to hop in on that one? Like, how are we going to get the the resources to do that?
I'll hop in.
Yeah.
I think one thing that we've heard a lot, and it's certainly true, cryopreservation is really challenging. But I think also just get started. Something small, you've got a laminar flow cabinet, you've got a small dewar, you start experimenting on it. And because a big part of cryopreservation is also upskilling, so just understanding how things work. So don't be put off if you don't have the millions to do it. I mean, we started with less than $10,000 to do the first thing. So don't be too put off to take a chance if you can. If you've got the resources, then just start and learn, and then reach out to the great people we've chatted to here, and you'll quickly learn how to do it. So
that's good advice, Karen.
Excellent. So look, I might just look at a few of the questions that have come in from the audience. So there's a question just on that. It's from Justin Collette, and he's saying, well, he's stating, well, presumably that the storage of exceptional species is more costly than orthodox. It's probably generally true. Do we have trouble with convincing the people who fund us about the needs for these species? And if we're having trouble, what can we do about it? Maybe Linda or Damian?
I'm happy to jump in there, Linda.
Mm-hmm.
Look, I think in Australia we've been really lucky with the example this morning from the Threatened Species Commissioner with the last Threatened Species Strategy and now the next one. I think they're really open to the conversation about where we are with plant conservation and seed science, and they understand that this is really, really difficult and it does take resources and time. I think it's a long-term discussion and educating funders about what's happening. Things like the germplasm guidelines and the Ian Potter Foundation funding that. It's a fantastic thing to see that funders are open to these ideas, and they're starting to, I guess, I'm noticing that funders are moving away from the very forward-facing eyes and coming around to the plant point of view, which I think is a great thing.
Thanks, Damian.
Just happy to add something there, Cathy.
Yes, please.
I think the question is not how expensive is it to conserve it, it's how expensive is it if we've lost it. And I think that's a conversation we rarely have, is what's the detrimental effect of losing a species? And it's actually significant. You might go, I've got plenty of redundancy in the system. But, Australia's got a unique heritage, as has PNG, as has New Zealand, that's not found very much anywhere else in the world, if at all. And it's our responsibility to maintain all those species. So, there is an ethical imperative that we do put the resources into it. I also think Karen's exactly right. You start small and things get cheaper over time. I look at the cost of genomic research, couldn't afford to do more than one species at a time. Now we're doing 20 or 30 or 100. So the costs of these things often come down over time, but if you can't demonstrate that you can do it, you're never going to get any investment.
Yeah, good message. Yeah, and we just have to keep taking that up with them. Yeah, thanks, Linda. There's so many good questions coming in. I'm not sure I'm going to get to all of them, but I will just go to Bryn in WA. Talking about pollen collection that Karen was mentioning before, do we know of any institutions in Australia who are storing pollen as another source of genetic diversity? It's just, I'm just putting that question out there so I can set it like it's a something that we do rarely in our organisation, and it's usually for a particular purpose. But is there somewhere in CSIRO that does that you're aware of? No. Okay. Thanks, Bryn.
I think we need just was going to comment, maybe we need our ag or forestry colleagues on the call. I think that might be the place where that sort of stuff's happening more.
Sal's online. She can comment in the chat.
that'd be great.
One of our agricultural colleagues would comment on that in the chat. From Chaminda, the ecosystems on Earth are changing day by day, and habitats and plants are being destroyed. Climate's changing, future climate will be different. So although we have preserved the plant species which are threatened, we'll be able to reintroduce them into the environments that have changed in the future, because the habitats will have changed, the climate will have changed. Any thoughts on that? You know, what we're preserving today may not be, we might need to find another place for it to grow. I think we might be possibly thinking about assisted migration here.
Really good question. That's something we've been working on in a project with ANBG and the Australian Tropical Herbarium, looking at trying to preserve species that are growing on tropical mountaintops up there. And there are 70 endemic species that are at real risk from climate change because they grow in cloud forest, and warmer climate is going to raise the cloud level, which is going to reduce the amount of water going into the environment and increase the amount of light, and that's going to have some really detrimental effects on those species. So we've been collecting the species and propagating them, or collecting seed when we can, and we've been distributing them to amongst the project partners, so to different botanic gardens that have a climate that's suitable for those species. And it might be that at some stage that habit isn't going to be suitable for them to grow at all anymore. So at least we will have those species preserved in some way.
Excellent. Yeah, that's a really great example, Karen, of plants with nowhere to go. What are we going to do with them?
Yeah.
Okay. I've got a question here from Chow Koon, I think from Singapore. Have there been any examples of species that have been taken from ex situ conservation and re-established back in natural environment and done that successfully? If that's so, could we learn from that for other threatened species? I think that might be one for you, Nathan.
Get myself off mute. Yeah, so a good example of that was the project that I was lucky to be involved with, which was the recovery program of the North Rothbury Pesunia, Pesunia pauciflora, which is a critically endangered species. I think the distribution of that species was about a 4.3 linear kilometre distribution, so very, very small. So we established that recovery program to learn more about its ecological requirements, but also to do iterative translocations concurrently to that research to understand more about how or whether we can actually translocate it, whether it'll survive and persist in the landscape. So we had that approach. We had a lot of success putting back plants, both propagated from seed and from vegetative cuttings and tissue culture. And we successfully, I think, planted just under 1,000 plants to bolster, I think, about the 400, 500 plants that were naturally occurring in its distribution. And that did include a couple of assisted migrations just outside of its distribution, and those plants are thriving well to this day. So yeah, it is possible.
I want to get this question in quickly, and then I want to go around and sort of finish up with a question to all of the panel. But from Justin Malley, he's asking, I think you're in the Northern Rivers region of New South Wales, if rainforest seedlings that survive in the understory, whether there's been much work done on germinating rainforest seeds and keeping them alive as seedling collections. So maybe Karen, if you or someone, Karen, maybe you could quickly answer that.
Yeah, it's not something that we do at the plant bank, but it is something that some nurseries do, like Firewheel Nursery in northern New South Wales. And they will germinate the seeds, put them into seedling trays, and then just reduce the amount of nutrients and hold them under shade. And they can keep them alive that way, not indefinitely, but they can keep them alive for 1 to 2 years and maybe 3 years, depending on the species. So it is a good way to, you know, rainforests often do this horrible thing where they'll all fruit at once and then they won't do anything for a few years. So if you're wanting to spread out your material for translocation over a period of time, then it's a good way to do that. It's not useful for long-term storage, but definitely useful for that application.
Awesome, thanks, Karen. All right, so quickly, I've got to wind up now. So that's been a really useful discussion. So many more questions that I didn't get to, and I apologise for that at this point. But the last question I have for each of the panelists, just in 1 or 2 sentences, if you could just tell me what you'd like to see happen in the next 5 to 10 years in this space. With Gibson, we'll go to you first.
In the next 10 to 5 to 10 years time, I would like to involve the locals, because in PNG we do have like locals are the owners, it's not the government. So that's our big challenge, to have access to the communities with the forest, and also to tie those skills and knowledge for projects with university students, engaging students to be involved, so that in the next 10 years time, we have somebody prepared for continuation of the work. So since I'm at the university, so I'm trying to look at tying the students at the universities to be involved with the communities, and getting some of these gem classroom establishment of where to store the seeds.
Yeah.
And
research with students is
yes.
Essential. That's great. Thanks, Gibson.
Yes.
Damian, where would you like to see it going?
Oh, look, I guess I'd like to see the conversation continue, and obviously see you all again in Horsham in 2025, and hopefully again 4 years later somewhere else, you know, whether that's in Australia or some other country in the region. I think just being able to see people's capacity, expertise, and knowledge improve, and seeing more of these species being brought back into banks and back out again into, you know, restoration or translocations, whatever it might be. But I guess just a more sharing of knowledge and expertise so we can all benefit.
Thanks, Damian. Karin?
Yeah, I think I will take from Damian and Gibson as well, and just see a significant increase in the people working in it, but don't underestimate the sharing as well, and getting students. So in New Zealand, it's not used very often, seed banking, and definitely not exceptional species. I really would like to see it becoming standard practice in at least some of the main gardens.
Right. Karen?
Yeah, I'll second that. It would be really great to have more people working in this area, and just, you know, sorting out the exceptional from the non-exceptional in the rainforest, and banking what we can, and then working harder on what we can't bank. And yeah, just more collaboration among people who have to deal with those sorts of species, in particularly in tropical and subtropical areas, who have those habitats that have a much higher proportion of them.
Excellent, thanks. Lydia?
I think along similar lines to the others, more people involved, but I guess in a more general sense, just a greater appreciation of what ex situ conservation methods can do. And I think not just that storage, but the associated research that we can then do with those plant materials that we bring into conservation. I think that's where we learn a lot about species and what we can and can't do for them. And so just general appreciation of that value of those collections and the associated research would be amazing to see.
Excellent. Thank you, Lydia. Nathan?
Yeah, just echoing what everyone else has kind of said. I'd like to see the research and conversation still continue. Certainly more cohesion between different groups, locals, communities, public, private sector. to achieve common goals, and certainly involving all the many relevant groups that are a part of these projects, not just the researchers, but also the horticulturists and restorationists as well. I think that would be great. Thanks,
Nathan. I'll give the last word to you, Linda.
Thanks, Cathy. All that, and at the 10-year horizon, I'd like to see a really strong, cohesive regional network that's working together, providing people the opportunity to move to other areas to learn, is engaging with the community, is engaging with policy, and is engaging with funding bodies to actually have a really strong, vibrant regional sector.
Thank you. Well, thank you to all the panelists. I'm going to hand back to Amelia now.
Thank you, Cathy. Thank you, Karen, and thank you to all our panelists this afternoon. And a big thank you to everyone who's attending and has contributed to the chat and has been answering questions posed by other attendees. It's wonderful to see that information sharing. Thank you. So on behalf of the Australian Network for Plant Conservation, I'd like to thank all the panelists and presenters for making time to share their experiences today. We appreciate the funding support of the Australian Academy of Science for today's Fenner Conference on the Environment. And a big thank you to all our support staff behind the scenes, including Lucy, Amy and Katie, and everyone who's helped us prepare for today, including the Seed Science Conference Organising Committee. I hope you've enjoyed the discussions today, and we look forward to seeing you at events run by the Australian Network for Plant Conservation in the future. Please stay online for a few minutes to find out how you can get connected to the ANPC and how we can help you share your important work on plant conservation. Thanks, everyone.